Hey, everybody. So, we've just talked about two different types of drugs that affect acetylcholine release in the peripheral nervous system. Now, we're going to talk about particular drugs that affect breakdown of acetylcholine. So, in particular, there are two different ways that neurotransmitters can be broken down. One of them is a process known as reuptake, which we'll talk about soon, and the other process is a breakdown by a particular enzyme. So, in this particular case, we have an enzyme called acetylcholine esterase, and the general rule is if it ends with the word ase, it's probably an enzyme. So, acetylcholine esterase actually exists in the synapse, in the synaptic cleft, and its job is to basically break down molecules of acetylcholine into choline, which serves as a precursor, and acetate. Once these are broken apart, they can actually be taken back into the presynaptic cell. So as you can kind of see in this figure, you see that choline is being taken back up into the cell by a transporter protein. It can be reused, recycled, and combined with more acetate. And so it's enzymatic degradation or breakdown by enzymes. That is what acetylcholine esterase does. And it turns out that there are different types of drugs that will inhibit the release, that will inhibit acetylcholine esterase, which basically makes it an agonist. If you don't have this enzyme, acetylcholine will not be broken down and it will exist in the synapse for a longer period of time. So acetylcholine esterase is important in both central nervous system pathways as well as the neurons of the peripheral nervous system. These drugs will affect acetylcholine by blocking acetylcholine esterase, and by doing so, they allow acetylcholine to remain intact longer than normal. So for example, we have donezepil, which is sold by the brand named Aricept. This is typically used to treat the early stages of Alzheimer's disease. Pesticides actually do the same. They block acetylcholine esterase, and they basically kill insects by paralyzing their breathing. And then we have sarin. This was originally developed as a pesticide by the Nazis. It is a UN classified weapon of mass destruction. It was used by Iraq against Iran in the 1980s. And basically, this will almost immediately paralyze respiratory muscles. So all of these function as agonists. Now, there are some receptors for acetylcholine, and it turns out that there are two different types. So we have one that is called nicotinic because they're stimulated by nicotine. And then we have muscarinic, which is stimulated by muscarine, which is a poison that is present in certain species of mushrooms. Nicotinic is what we call an ionotropic receptor, and muscarinic is what we call a metabotropic receptor. So ionotropic receptors, basically when nicotine binds to this receptor, that allows the flow of ions to come in and out of the cell. And it's a pretty fast-acting receptor. Muscarinic, when muscarine binds to this receptor, because it's metabotropic, it's going to trigger a variety of different cellular changes, and it tends to be slower acting. So nicotine is a direct agonist, and it enhances acetylcholine, and muscarine is also an agonist. So nicotinic receptors are largely present in the central nervous system and also in the somatic nervous system, and they're generally excitatory and fire excitatory postsynaptic potentials. Muscarinic receptors are present throughout the central nervous system and the parasympathetic nervous system, and they fire both EPSPs, so excitatory postsynaptic potentials, and inhibitory postsynaptic potentials. So let's talk about some of the different drugs that affect acetylcholine receptors. All of these are drugs that are found in nature. So as I've mentioned, nicotine is an agonist. It's actually found in tobacco. It's also found in certain species of eggplants, peppers, coca plants, where you can also find cocaine. And largely, it basically increases sympathetic effects. So it increases heart rate. It helps you feel more alert. It constricts the blood vessels. It can produce a bit of a muscle twitch. Now, muscarine does not cross the blood-brain barrier, so it only acts on the peripheral nervous system, in particular the parasympathetic nervous system. So these effects will include things like salivation and vomiting. If you eat poison mushrooms, death is very rare, although the effects are obviously pretty unpleasant. Now, curare is a direct antagonist on the nicotinic receptors, and so it blocks acetylcholine. It's found in plants native to South America and is used for poison darts by native peoples there. It also does have some medical uses besides paralyzing prey. It's used to relax skeletal muscles during surgery. It does not cross the blood-brain barrier, so it largely affects the nicotinic receptors of the skeletal muscles. And then atropine is a direct antagonist of the muscarinic receptors in both the peripheral or parasympathetic and the central nervous system because it does cross the blood-brain barrier. It's found in many species of plants, which we'll talk about soon. It does have the opposite effect of muscarine, so that will be things like reduced salivation and reduction in other secretions that promote digestion. It can also produce hallucinations due to its effects on the central nervous system. It does have some interesting medical uses, such as treatment of bradycardia, which is having a slow heart rate. So here are images of the Aguas natives from Peru utilizing curare tip darts to help paralyze prey. In addition, some other plants that are often used that contain atropine are typically called members of the Nightshade family. This is a family of plants containing atropines. It's been used since about the 4th century as a supposed love potion. It produces dream-like hallucinations. One of the cholinergic pathways, as I mentioned before, has a role in sleep-wake behavior. Atropine will cross the blood-brain barrier. It blocks the muscarinic receptors, so it causes sleepiness and a dreamlike state. It is also used to dilate pupils. So for those of you that know Shakespeare, there is a quote from Antony and Cleopatra. Here is a mandrake fruit. If you're a fan of Harry Potter, here's what they actually look like. So now we're going to talk about a pretty important group of neurotransmitters. And as you've kind of noticed, we do classify neurotransmitters on the basis of their molecular structure. So we're going to talk about a group known as the monoamines. That includes dopamine, serotonin, and norepinephrine. So monoamines are called so because they contain one amine or amino group. And as you're going to see on the following slide, they have a really similar molecular structure. And what this means is that because they have such a similar structure, many drugs will affect more than one of these neurotransmitter systems. So a drug might affect dopamine and norepinephrine, or dopamine and serotonin. We can basically subdivide the monoamines into 2 different groups, one group called the catecholamines and one group called the indolamines. We will discuss 3 catecholamines, as you can see, dopamine, epinephrine, and norepinephrine, and one indolamine known as serotonin. They do not cross the blood-brain barrier. So what this means is that we use precursors to help synthesize these neurotransmitters in the neurons. We can't administer dopamine or serotonin as a medicine. They have to be given a precursor or something that does cross the blood-brain barrier that actually will enhance or, in certain cases, inhibit these. So first, we're going to talk about the catecholamines. They have very similar structures with just minor differences. The reason that these are called catecholamines is because every single one of them contains a catechol ring. I'm just showing you this so you can see the similarities. You will not have to draw these on a test. So dopamine is produced by neurons in the midbrain. It's involved in numerous functions, including things like movement. It's also pretty critical for addiction to drugs. Norepinephrine and epinephrine are very similar in function. Both of these are produced in the adrenal gland. They sit above the kidneys and they receive input from the autonomic nervous system. Both are also produced in the pons region of the brainstem in an area called the locus coeruleus. Both of these will result in increased heart rate and vigilance, as well as constricted blood vessels. Norepinephrine is also known as noradrenaline, and epinephrine is sometimes known as adrenaline. Nor is a German prefix for without. We call it norepinephrine because, unlike epinephrine, it does not contain a methyl group. So here's how the catecholamines are synthesized. This is in your book, so hopefully you should be able to follow along. It shows the relatively simple and small differences between the catecholamines and the enzymes that are used to convert one to another. So the ultimate precursor to both dopamine and norepinephrine is tyrosine. Tyrosine is an amino acid that we primarily obtain from meat and cheese. Tyrosine is then converted into L-DOPA, which is a precursor to dopamine. L-DOPA is so effective as a dopamine agonist that we use it to treat diseases where dopamine is depleted, like Parkinson's. L-DOPA is then converted into DOPA and in dopamine. And so what you can kind of see is that the differences between these neurotransmitters are really small, which are highlighted by the circles in purple. So these are some pretty small differences. Here is a much simpler version. This also shows epinephrine, which is a simple derivative of norepinephrine. So let's start by talking about each of the monoamines in more detail. We're going to start by talking about dopamine, which has many important functions. Dopamine has 4 important pathways in the brain that are associated with the majority of the effects. So we first have what is called the nigrostriatal tract. This basically connects the area of the midbrain known as the substantia nigra in the midbrain with the striatum or the basal ganglia right here. This area is really important for movement. A very critical tract that we're going to talk about, or a pathway, is the mesolimbic pathway. This connects the ventral tegmentum of the midbrain to the nucleus accumbens, the amygdala, and the hippocampus of the limbic system. This area is really important for reward and addiction to certain behaviors, including drug use. So this is what we occasionally refer to as the addiction pathway. We have the mesocortical tract, which connects the ventral tegmentum to the prefrontal cortex. This area is really critical for planning, memory, attention, and problem solving, and to a lesser extent, addiction and reward. And then finally, we have the tuberoinfundibular tract. This connects the tubular region of the hypothalamus to the infundibular region of the hypothalamus, and this helps control hormone secretion in the hypothalamus. As with the acetylcholine pathways that we've talked about already, this figure is meant to show the complexity of these systems. Multiple regions are often affected by a single neurotransmitter, and many effects can be produced. And this is one issue anytime that you take a drug. It may have one desired effect, but you have a lot of other effects that get produced as a result.